Joint hypermobility. Joint hypermobility, often called being double-jointed, describes an increased range of motion and flexibility in joints beyond what most people can achieve. This trait has a strong genetic component, with heritability estimated at 70% based on twin studies. The mutation comes from multiple genes that encode type 5 collagen, an important component that gives connective tissue its strength and structure. Variations in these genes can alter how much tension joints naturally maintain and increase flexibility. In certain physical activities, this provided advantages. General joint hypermobility is surprisingly common, affecting anywhere from 2 to 57% of the population, depending on age, sex, and ethnicity. Studies in adult female twins show prevalence rates of 19.5 to 22%, indicating roughly 1 in 5 women has notable hypermobility. The condition is more common in females than males, possibly due to hormonal effects on connective tissue laxity. Prevalence declines with age, falling from 34% in people aged 20 to 30 years to just 18% in those aged 60 and older. This age-related decrease likely reflects gradual stiffening of connective tissues over time. Ethnicity also influences rates, with some populations showing higher baseline flexibility than others. The advantage of hypermobility lies in enhanced physical performance for activities requiring extreme flexibility. Dancers, gymnasts, contortionists, martial artists, and other performers benefit enormously from joints that naturally extend beyond normal ranges. Hypermobility allows movements that would injure less flexible individuals, enabling complex poses, deep stretches, and unusual body positions. However, hypermobility comes with significant trade-offs. Joints that move too freely lack stability, increasing the risk of dislocations, sprains, and chronic pain. Loose ligaments cannot hold bones in optimal alignment under stress, allowing abnormal movements that damage cartilage and surrounding tissues over time. Many hypermobile individuals experience joint pain, fatigue, and instability that limits physical activities. The trait also correlates with higher rates of anxiety and autonomic dysfunction, possibly due to shared genetic influences on connective tissue throughout the body, including blood vessels. About 10% of people with general hypermobility develop significant physical or psychological symptoms. Dry earwax. Earwax exists in 2 distinct types, wet and dry. This difference results from a single nucleotide mutation in the ABCC11 gene on chromosome 16, with A replacing G at position 538, altering the protein produced. The altered protein shows reduced function in transporting lipid molecules across cell membranes, including the components that make earwax moist and sticky. Individuals with this mutation produce dry, flaky earwax that crumbles easily. On the other hand, those with the normal protein produce the wet, sticky wax type most people consider normal. The same normal gene also controls the sweat gland secretion, directly linking earwax type to body odor. The mutation arose in Northeast Asia, likely in ancient China approximately 2,000 generations ago. Genetic analysis of the Tianyuan man from northern China, who lived 40,000 years ago, shows the variant was already present, indicating remarkable continuity. The mutation then spread across East Asia before reaching other regions through migration. The distribution follows a clear geographical gradient, highest in northern China and Korea, decreasing toward southern Asia, and dropping sharply westward toward Europe. The evolutionary advantage in cold climates was better thermal regulation. Wet earwax contains higher water content, which could freeze in frigid environments, causing discomfort or damage to the ear canals, while dry earwax remains stable. Since earwax and the apocrine sweat glands were associated, dry earwax also meant less apocrine sweat gland activity, which causes minimal body odor. In ice-bound conditions where bathing was difficult or dangerous, lower apocrine secretion also reduced the need for frequent washing. These physiological adjustments helped ancient populations survive harsh northern winters. However, the dry earwax phenotype comes with other effects. The same mutation has been linked to reduced colostrum secretion in postpartum women. Studies show 67% of women with dry earwax experienced absent colostrum production compared to 40% of wet earwax women. This colostrum is the early yellowish milk that is rich in nutrients and antibodies and crucial for newborns. Dimples. Dimples are small indentations that appear in the cheeks or chin when smiling, created by variations in facial muscle structure. Cheek dimples, the most common type, result from a shorter or differently attached zygomaticus major muscle, which runs from the cheekbone to the corner of the mouth. When this muscle contracts during smiling, the skin above it gets pulled inward instead of simply lifting upward. This creates the characteristic depression. Chin dimples form through a similar mechanism involving the mentalis muscle. Dimples are relatively common, appearing in approximately 20 to 30% of the population, though exact exact frequencies vary by ancestry. They are inherited as a dominant trait, which means they can appear even when only one parent carries the variant. However, the genetics are not completely straightforward. Some individuals with dimples have children without them, and vice versa, suggesting multiple genes or incomplete penetrance influence the trait. Dimples can also appear asymmetrically, with one cheek showing a dimple while the other does not. This asymmetry reflects developmental variation in muscle attachment during fetal growth. The evolutionary origin of dimples likely has no adaptive significance beyond social perception. They appear to be a neutral anatomical variation that became preserved simply because they caused no harm and may have offered minor social advantages. In ancestral environments, facial expressions conveyed critical information about emotional state and intent. Dimples enhance the visual impact of smiling, making the expression more noticeable and memorable. This may have facilitated social bonding, particularly in mate selection and social hierarchies. Features that increased perceived friendliness or attractiveness could spread through sexual selection, even without direct survival benefits. The advantage of dimples is purely social and perceptual. Research in social psychology consistently shows that faces with dimples are rated as more approachable, friendly, and attractive across cultures. The indentations draw attention to the smile, making positive expressions more prominent. This creates a halo effect where dimpled individuals are perceived more favorably in social interactions, potentially benefiting from increased trust, likability, and social bonding. In evolutionary terms, traits that enhanced mate attractiveness or social acceptance could spread through populations even without improving survival directly. Dimples represent this category of features. Cilantro tastes like soap. For some people, cilantro tastes unmistakably like soap, metal, or dirt, rather than the fresh citrusy herb most others enjoy. This polarizing perception stems from variations in the OR6A2 gene, which encodes a taste receptor that detects aldehyde compounds. For people with this mutation, their OR6A2 receptors bind more strongly to the aldehydes present in cilantro leaves, triggering a perception of soapy or offensive flavors instead of pleasant ones. The difference is purely sensory, not psychological preference. Global rates of cilantro aversion vary dramatically by ancestry, revealing both genetic and cultural influences. Among people of East Asian descent, approximately 21% report finding cilantro genuinely offensive, the highest rate worldwide. Europeans follow at 17%, while those of African descent show 14% aversion. The numbers drop sharply in populations where cilantro features prominently in traditional cuisine. South Asians report only 7% dislike. Hispanic populations show 4%, and Middle Eastern populations have the lowest rate at just 3%. These patterns suggest that repeated exposure and cultural context can influence whether genetic predisposition translates into actual aversion. The evolutionary advantage of heightened aldehyde sensitivity was toxin avoidance. Many poisonous plants produce aldehydes and related compounds that signal danger through bitter or offensive tastes and smells. Individuals whose OR6A2 variants detect these chemicals at lower concentrations would instinctively avoid potentially harmful vegetation. In ancestral environments without botanical expertise or written knowledge, this genetic alarm system prevented poisonings. Children especially benefited, as their inexperience made them vulnerable to consuming dangerous plants. The cost was simply missing out on one particular herb that happened to trigger the same receptors. However, the heritability of cilantro aversion is surprisingly low. Genetic factors explain only about 8.7% of the variation in whether someone finds cilantro soapy or pleasant. This means environmental factors, repeated exposure, and cultural context play substantially larger roles than genes alone. Many people with the genetic variants associated with aversion still enjoy cilantro, while some without those variants find it unpleasant. Psychological associations and learned preferences all contribute significantly to the final perception. Blue eyes. No actual blue pigment exists in the iris. Blue eyes result from a mutation in the switch controlling the OCA2 gene's expression, which is a protein involved in melanin production in the iris. The mutation reduces OCA2 activity, limiting melanin production in the front layers of the iris. With very little melanin present, light entering the eye scatters according to wavelength. Shorter blue wavelengths scatter outward, reflecting back to observers. Genetic analysis reveals that every person with blue eyes today inherited the same ancestral variant, making all blue-eyed individuals distant cousins through this single shared ancestor. Blue eyes are most common in Northern and Eastern Europe, particularly in the Baltic states, Scandinavia, and Iceland, where frequencies can exceed 80%. In Ireland, Scotland, and Germany, rates range from 40 to 60%. The frequency declines moving southward and eastward, dropping below 10% in Southern Europe, the Middle East, and Central Asia. Blue eyes are exceedingly rare in East Asian, African, and indigenous American populations, where the mutation never spread. Globally, approximately 8 to 10% of people have blue eyes, making it a relatively uncommon trait concentrated in specific ancestral populations. The evolutionary advantage, while it remains debated, is enhanced low-light vision. With less melanin-absorbing light in the iris, more light rays reach the retina, potentially improving visual sensitivity in dim conditions. This could have benefited hunters, gatherers, and early farmers operating during dawn, dusk, and overcast days common in northern climates. Alternatively, blue eyes may have spread through sexual selection, where the novelty and distinctiveness of the trait made individuals more attractive as mates. Traits that stand out visually can become favored in mate choice, even without survival benefits, particularly in small populations where rare features become desirable. However, blue eyes come with clear disadvantages. Reduced melanin provides far less protection against ultraviolet radiation, making blue-eyed individuals more susceptible to light sensitivity, glare, and potential damage from prolonged sun exposure. The lack of melanin also means blue eyes cannot adapt to varying light conditions as effectively as darker eyes, which can adjust pigment density somewhat. These vulnerabilities explain why blue eyes remained concentrated in northern latitudes with weaker sunlight and never became common in sun-intense equatorial regions. Red hair. Red hair emerges from mutations in the MC1R gene on chromosome 16, which controls melanin production in hair and skin. Individuals with both copies of this gene variant produce primarily pheomelanin instead of the darker protective eumelanin. This shifts pigmentation from brown or black to the distinctive copper, auburn, or bright red tones that define redheads. The gene variants are recessive, meaning both parents must carry at least one copy for a child to express red hair, though parents themselves may have different hair colors if they are heterozygous carriers. So, parents that carry one copy each and without red hair can still produce a redhead. These mutations likely arose in Northern Europe, where reduced sunlight diminished the selective pressure for dark, UV protective pigmentation. Initially thought to be an adaptation for better vitamin D synthesis in low-light environments, more recent evidence suggests the prevalence of the gene variant spread through genetic drift rather than strong selection. As humans migrated from sun-exposed Africa to cloudier northern climates, maintaining high eumelanin levels became less critical for survival. Red hair is exceptionally rare globally, found in only 1 to 2% of the population. The highest concentrations appear in Scotland, with roughly 13% of people carrying the trait, followed by Ireland at 10%. Rates drop dramatically outside northwestern Europe, approaching near zero in East Asian, African, and Native American populations. The evolutionary advantage of red hair itself may be minimal, but the associated fair skin allows for rapid vitamin D production in weak sunlight. Vitamin D is critical for bone health, immune function, and calcium absorption. In northern latitudes, where UV radiation is scarce for much of the year, lighter skin enabled sufficient vitamin D synthesis even during short winter days. This adaptation supported skeletal strength and reduced risk of deficiency-related diseases like rickets, which weaken bones. The trade-offs, however, are substantial. The red hair pigment offers far less protection against UV radiation than eumelanin, resulting in dramatically increased skin cancer risk. Redheads burn more easily and more intensely, and studies show they face significantly higher melanoma rates even with identical sun exposure compared to darker individuals. These vulnerabilities explain why red hair remains rare outside regions with minimal year-round sun intensity. In equatorial or high UV environments, the trait would be severely selected against. Alcohol flush reaction. Alcohol flush reaction turns your face bright red within minutes of drinking, accompanied by a racing heart, nausea, and headache. This dramatic response comes from a single letter change in the ALDH2 gene on chromosome 12, where a G becomes an A at position 504. The mutation changes an enzyme structure, crippling its ability to break down acetaldehyde, the toxic compound your liver produces when breaking down alcohol. Without the functional ALDH2 gene, acetaldehyde accumulates rapidly in your blood, triggering the flush and making even small amounts of alcohol intensely unpleasant. Most people think rare genetic mutations are random quirks or strange leftovers or useless biological accidents, but human evolution doesn't keep traits that don't serve a purpose. Many of the rare features you carry today survived because they quietly helped your ancestors adapt, survive, attract partners, or sense the world differently. The mutation responsible for the alcohol flush reaction originated in ancient China and spread across East Asia over the past 40,000 years. Today, approximately 540 million people worldwide carry at least one copy of the ALDH2 variant, representing 8% of the global population. In East Asian populations, the numbers climb dramatically. Between 30 and 50% of Chinese, Japanese, and Koreans experience the flushing response. Among Koreans specifically, studies show 40% of the population has alcohol flush reaction, with the highest rates appearing in people aged 60 to 69. The mutation is virtually absent in European and African populations, though recent research identified rare ALDH2 variants affecting an estimated 120 million people of non-East Asian ancestry. The advantage of this mutation is behavioral protection against alcoholism. The immediate, visible, and physically uncomfortable response to alcohol creates a powerful deterrent against heavy drinking. Studies consistently demonstrate that individuals with ALDH2 deficiency show dramatically lower rates of alcohol dependence. The people who experience the most severe reactions are almost never found among alcohol-dependent populations. Even those with milder symptoms consume significantly less alcohol than those with fully functional enzymes. This built-in biological brake protects against addiction, liver disease, and the cascade of health problems associated with chronic drinking. However, the mutation carries serious risks for those who drink despite the warning signals. ALDH2 deficient individuals who continue consuming alcohol face 4 to 8 times the risk of esophageal cancer compared to those with normal enzyme function. The prolonged exposure to acetaldehyde in saliva and tissues damages DNA directly, promoting tumor formation. Additional risks include elevated rates of stroke and cardiovascular disease when alcohol consumption continues regularly. The protective effect against alcoholism paradoxically places some carriers at extreme cancer risk if they override their body's biological alarm system and maintain drinking habits. Human evolution continues. Every generation brings new mutations, and changing environments create new selective pressures. Some will increase in frequency as conditions change. Others will decline or disappear. But all exist today because they offered advantages to someone, somewhere, at some point in the deep past. They are your evolutionary inheritance from your ancestors, written in DNA, expressed in traits, and passed to future generations. Have you discovered any unexpected genetic traits in yourself? Do you have the alcohol flush reaction, hate cilantro, have dry earwax, or carry any of these hidden advantages? Let us know in the comments and subscribe for more videos exploring the evolutionary stories written in your genes.